Welcome back to the deep dive. I want to start today with a question that, it feels like it belongs in a philosophy class, but we are actually going to answer it with some pretty hard biology.
Okay.
The question is, what is it specifically that makes the human brain human?
That is the ultimate question, isn't it? And if you just look at the raw stats, it's actually a really confusing one.
Right, because I think the common assumption is just size. We have big heads, so we have big brains, and therefore we're smart. But that doesn't really hold up, does it?
Not at all. I mean, if intelligence were just about brain weight, elephants and sperm whales would be running Wall Street.
They have much larger brains.
It's much larger. And even Neanderthals had brains that were, roughly the same size as ours, sometimes even a little bigger.
So if it's not just the volume, the next place people usually go is the wiring. And for decades, the story I've always heard is that humans are just great at excitation. We have these huge highways of neurons sending go signals all over the brain.
That's the classic narrative, the projection neuron theory. The whole idea was that humans just developed these superhighways to send information faster and farther than any other species.
Right.
We thought we were smarter because our neurons could basically shout louder.
But the research we're diving into today, specifically a commentary by Bonafina and Nguyen on a really groundbreaking study by Glia and their colleagues, it suggests we might have been looking at the wrong half of the equation entirely.
We absolutely were. It turns out the secret to human complexity might not be the ability to shout.
What is it then?
It might be the ability to whisper, or maybe more accurately, the ability to say hush.
We're talking about inhibition, the brakes.
We are, and we're going to look at a brand new discovery, a very specific type of cell factory in the human brain that seems to be primate-specific, and its entire job is building this massive, complex fleet of quieting cells.
And the mechanism it uses to do this is, well, it honestly sounds like science fiction. You were telling me the cell doesn't just divide, it physically jumps, it launches itself.
It's an incredible piece of biological engineering, but, and this is the part we really have to get to, this complexity comes with a price. The source material draws a direct genetic line between this fancy jumping cell and autism spectrum disorder.
So the very thing that makes us complex is also what makes us vulnerable.
That's exactly it.
Okay, let's unpack all of this. We have to start with the landscape. If I could, take the top off a skull and look at a human brain versus, say, a mouse brain, what's the most obvious visual difference?
The wrinkles. The human brain looks like a walnut. A mouse brain is smooth, kind of like a grape.
And the source material calls this pronounced gyrification.
Which is just the $10 word for folding. But it's really important to understand why it folds. It's a physics problem, really.
How so?
As humans evolved, we needed more surface area, more cortex to run all our complex software. But we have a hard limit, the size of the birth canal.
Right. If the skull gets any bigger, you can't be born.
Exactly. So nature's solution was to crumple up the paper. You take this giant sheet of cortex and you just fold it over and over and over so it fits inside the jar. That's gyrification. A morphogenetic adaptation to maximize processing power in a confined space.
Okay, so we have a folded brain, and inside those folds we have these two teams of neurons we hinted at, the excitatory and the inhibitory. I want to really nail down the difference here. You use the analogy shouting versus hushing. Can you give me another one?
Think of a symphony orchestra. The excitatory neurons are all the musicians playing the instruments.
Okay.
The violins, the horns, the drums, they're the ones producing the sound. They are the raw data.
Got it. So the inhibitory interneurons.
Yeah.
They're not musicians.
No, they're the conductor.
The conductor doesn't make a sound.
Right. But the conductor decides who plays and when. Without a conductor, if every single musician played their note as loud as they possibly could all at the same time, what would you have?
Just noise.
Yeah.
Total cacophony.
Yeah. Exactly. And in the brain, that noise is a seizure. If you have all go and no stop, the whole system crashes. You need that conductor to say, violins, quiet down, cellos, you come in now. That is what interneurons do. They carve the signal out of the noise.
And the source is saying that as humans evolved, we didn't just add more musicians?
We added way more conductors. The ratio changed. We have a relative increase in the proportion of interneurons compared to rodents. We basically scaled up the management layer.
And that lets you play more complex music.
It allows for what the paper calls circuit flexibility and higher computational power. It allows for nuance. I mean, learning, decision-making, regulating your emotions, all of that is about inhibition. It's about not reacting to every single thing, but selecting the right reaction.
Now, here's a detail in the text that I found kind of surprising. We didn't actually invent new types of conductors, did we?
No, we didn't. And that's the humbling part. We share the same cardinal classes of interneurons with mice and ferrets. Evolution didn't reinvent the wheel.
So we're just using the same parts list.
We're using the same basic chassis. But the text says we have marked expansion and enriched transcriptional diversity.
Can you translate that for us?
It means we took the basic model and we customized it. Like, imagine a mouse has a standard stop sign. Humans took that stop sign and turned it into a smart traffic system that can sense rain, adjust its own timing, and communicate with self-driving cars.
So it's the same basic function, stop, but the implementation is vastly more sophisticated.
Infinitely more.
Okay, let's pause here. I want to do a quick recap just to make sure we've got this foundational layer locked in, because I feel like everything else builds on this.
Sounds good.
So point one, the human brain folded up on itself to fit more processing power into our skulls. That's gerification.
Right.
Point two, to manage all that power, we didn't just add more signal. We disproportionately added more brakes, more inhibition.
More Gurk conductors, yep.
And point three, we took the standard brake cells found in other animals and just supercharged them into a high-tech version.
That is a perfect summary. You've got the landscape. So now we have to go look at the construction site.
Right. Where do these high-tech conductors even come from? Because they aren't born where they end up working, are they?
No, and this is fascinating. In the adult brain, they live up in the cortex, the outer layer. But they're born deep, deep, deep in the basement of the developing brain.
The text calls this place the ganglionic eminence.
The GE sounds like a secret society, doesn't it? The ganglionic eminence. But it's really just a specialized factory floor near the base of the fetal brain.
And this factory, it has different departments.
It does. The source goes into two main production lines, the medial GE, or MGE, and the caudal DE, the CGE. Think of them as the middle and the tail departments of the factory.
And they make different things.
They specialize. The MGE makes 2 specific classes of interneurons, and the CGE makes 3 others. But for our story, the story of what makes humans unique, we really have to focus on the human MGE, the HMGE.
Because the human version of this factory is different.
It's huge. The source says it has an expanded subventricular zone.
Subventricular just means below the ventricle, right? Below that fluid-filled space in the brain.
Correct. So you have this massive factory floor. But the real difference isn't just the space, it's the time. The text specifically mentions a prolonged period of neurogenesis.
How long are we talking?
In a mouse, this factory closes up shop pretty early in development. In humans, it stays open for business right through the 2nd and 3rd trimesters of pregnancy. We are building these crucial cells for months and months.
And inside this factory, you have these little VIP rooms. The text calls them DNs.
Dividing ependymal desks.
I love that name. It sounds so cozy. What's happening inside a D?
These are specific pockets where the stem cells gather to divide. But here's a detail that just blew my mind, and I bet most people have never heard this.
Okay.
They aren't alone in there. They have helpers.
The resident microglia.
Yes. Now, usually when we talk about microglia in neuroscience, what are they?
They're the cleanup crew, the janitors. They're the brain's immune system. They eat trash and dead cells.
Exactly. But here, in the developing human brain, the janitors are acting like midwives. The source says these microglia release insulin-like growth factor to help generate and support these new interneurons.
So the immune system is actively helping to build the brain.
It is a critical part of the construction crew. It just shows how collaborative and complex this whole process is. You have the factory, the VIP nests, and the support staff all working together to churn out these cells.
But who is the actual builder? Because this brings us to the big reveal of the paper. The new suspect.
The SVZ-RGC.
The subventricular zone radial glia cell. Let's stick with SVZ-RGC. Why is this particular cell the headline news?
2 huge reasons. First, the source says it appears to be a primate-specific cell class. Mice don't seem to have this exact worker. It's an upgrade we got on the evolutionary tree.
And the second reason?
It moves. And I don't just mean it drifts around. It performs a very specific, very active maneuver called mitotic somal translocation.
Okay, you dropped that term earlier, and I promised we'd unpack it. Mitotic somal translocation. Break that down for me word by word.
Sure. Mitotic means dividing. It's in the act of splitting into 2. Somal refers to the soma, which is the body of the cell where the nucleus is, and translocation just means moving from one location to another.
This dividing body moving.
Right. But you have to visualize what's happening. In a normal cell division, the parent cell kind of plants its feet, stays still, and splits. It's a stationary process.
Okay.
The SVZ-RGC is totally different. Before it divides, it sends out this long, fiber-like grappling hook, attaches it to a structure much further up, and then it hoists its entire body upward.
Like a pole vaulter or something.
Or like it's climbing a rope. It rapidly yanks its cell body up through the crowded factory floor, and then, once it reaches its new position, it divides.
That sounds incredibly energetic. Why would it go to all that trouble? Why not just divide where it is?
The theory is it's about overcrowding and positioning. Remember, the human factory, the HMGE, is huge, and it's packed with cells. This jump allows the progenitor to escape the crush down at the bottom and position itself in a specific layer where there's more space and resources to do its job properly.
So it's actively finding its spot on the assembly line.
Exactly. It's an active, dynamic process of seeking the right environment.
Okay, so once it gets there,
Yeah.
what does it make? Because this was the other big surprise in the text. We used to think these cells were specialists. I make neurons, you make glia.
Right. The old one progenitor, one fate dogma. But the SVZ-RGC just shatters that rule. It is multipotent.
Meaning it can make multiple things.
It's a generalist. The source specifically says these cells produce interneurons, or prefix cops, but they concurrently produce glia, specifically cortical astrocytes and oligodendrocyte progenitor cells, or OPCs.
And glia are the support cells, the insulation, the maintenance crew for the neurons.
Yes. So imagine a car factory. Usually you have one assembly line for the cars and a totally separate factory for the spare parts and the mechanics. This cell is producing the race car, the neuron, and the pit crew, the glia, on the exact same assembly line at the exact same time.
That feels very intentional.
It has to be. The source suggests they come from a shared pool. Think about what that implies. If the neuron and the cell that's designed to take care of that neuron are born from the same parent at the same moment in the same place.
They're probably a matched set.
Exactly. They're likely genetically programmed to work together. It ensures that when that neuron finally migrates all the way up to the cortex, it has a sibling cell there ready to support it.
And speaking of where they are born, the text mentions these spatial domains along the rostrocaudal axis.
Yeah.
That's just head to tail, right?
Yep, this is the idea that geography is destiny for these cells.
How so?
Well, the factory floor is basically laid out like a map. If an SVZ-RGC jumps to a spot near the head end of the factory, it produces one specific subtype of interneuron. If it jumps to a spot down at the tail end, it produces a totally different subtype.
So the cell doesn't just randomly decide what to become. It knows what to be based on where its parent was sitting.
Precisely. The incredible diversity of our brain cells is spatially encoded right from the start.
Okay, I need to do another check-in here because we've added a lot of complex layers. Let me see if I can stack them up correctly.
Let's hear it.
Layer 1. We've got this huge, long-lasting factory in the basement of the developing brain.
The HMGE.
Right, layer 2. We have a special primate-only worker, the SVZ-RGC, that uses a kind of grappling hook method to jump through the crowd.
Mitotic somal translocation.
Yeah, layer 3. When it finds its spot, it's a multitasker, building both the neurons and their support crew at the same time.
The race car and the pit crew.
And layer 4. Exactly what it builds depends on exactly where it landed on the factory map.
You are 2 for 2 on the summaries today. That right there is the mechanism of human complexity.
It sounds perfect. It sounds like a Swiss watch. But,
Right.
and I feel like there's always a but in biology. Complexity usually comes with a bill to pay.
It does, and in this case, the bill is steep.
The source material connects this specific cell, this jumping SVZ-RGC, to a very specific medical condition.
It connects it directly to autism spectrum disorder, or ASD.
How specific is that link? Is this just a guess?
No, it's genetic. The paper states that SVZ-RGCs and all their descendant lineages are enriched in autism spectrum disorder risk genes.
Enriched. So that means if you take all the genes that we know are linked to ASD and you look for where they're most active in the developing brain, they light up right in this cell.
X marks the spot.
Why here? Why this cell in particular?
Well, think about everything we just described. The jumping, the multitasking, the incredibly long timeline that goes right through the 3rd trimester, the complex migration these cells have to do later.
It's a lot of moving parts, a lot of things that could go wrong.
It's an incredibly delicate ballet. The text calls it a potential point of vulnerability. Because the process is so complex and it takes so long, there are simply more opportunities for something to go slightly wrong.
So if the jump fails, Or if the timing is off by just a little bit.
Then the interneuron doesn't get to the right spot in the cortex, or it doesn't mature correctly. And remember what these cells are. They're the traffic cops.
Right. And if the traffic cops don't show up to the intersection,
you get chaos. And the source links this kind of disruption not just to ASD, but also to epilepsy and schizophrenia.
Which makes perfect sense when you think about that excitatory-inhibitory balance we started with.
Exactly. Epilepsy is a classic failure of inhibition, too much go, uncontrolled firing. Schizophrenia involves disordered processing, a failure to filter out the noise. And ASD involves fundamentally altered connectivity and processing. They all trace back, in part, to how these inhibitory circuits are built.
It's a really profound realization. The argument here is that the very mechanism that gives us our human cognitive abilities, this massive, complex expansion of inhibition, is the exact same mechanism that creates the risk for these disorders.
It suggests they aren't just random mistakes in the system. They are an inherent byproduct of our evolution. You cannot have the cognitive height of a human brain without also having the risk of falling off that ladder. It's the price of admission.
That really reframes how we should think about neurodivergence. It's not a bug. It's woven deeply into the blueprint of what makes us human.
It really is.
Now, before we close out, the source leaves us with a bit of a cliffhanger, a big debate that hasn't been settled yet.
The debate about fate plasticity.
Fate plasticity. That sounds dramatic. What does it mean?
Well, we talked about geography as destiny, that the cell is programmed what to become back in the factory. But the big question is that programming totally locked in? Or does that young neuron still have some choice as it develops?
You mean as it's migrating from the basement all the way up to the cortex, can it change its mind?
Exactly. Is it listening to its environment? If it finds itself in a different neighborhood than it expected, does it adapt and become a slightly different type of neuron? Or is it just a robot that's simply following a pre-written card no matter what?
Why is this the big unanswered question? What's at stake?
The medical implications are enormous. If a cell's fate is 100% locked in at birth, then treating these disorders becomes incredibly hard. You'd have to, go back in time and fix the factory during development.
But if there is plasticity?
If there is plasticity, if the cells are still malleable as they travel, then maybe we can influence them. Maybe we can develop therapies that could guide a wandering neuron back to the right path or help it mature correctly, even if its start was a bit rocky.
So the next frontier of this science is really figuring out how much of our brain is hard-coded versus how much is soft-coded.
Nature versus nurture playing out at the level of a single cell.
Incredible. When I started this deep dive, I wanted to know what makes us smart. I honestly expected you to tell me about a bigger engine.
And instead, I told you about better brakes.
It's a powerful takeaway. We live in a world that is constantly pushing for more output, more data, more speed. But our own biology is telling us that true intelligence, true sophistication, comes from the ability to inhibit, to regulate, to choose what not to do.
I think that's right. Maybe the smartest thing your brain does isn't the one thought you have, but the 1,000 other thoughts you suppress so that the one good one can finally shine through.
That is something I'm going to be thinking about all day. And for you listening, knowing that your intelligence relies so much on stopping rather than going change how you view your own mind? It certainly changed mine.
Stay curious.
Thanks for diving in with us. We'll see you next time.